It is easy to understand that whenever sound enters the ear canal conducts the sound to the eardrum, which we call as a tympanic membrane. Tympanic membrane, up to tympanic membrane is the external ear. From that membrane to the inner ear wall, it is a middle ear and then an inner ear. These are three portions of the ear. Function of the external sound is to collect the sound and transfer it on the tympanic membrane. Function of the tympanic membrane is to amplify the sound. There are three bones, all of you know, the malleus, incus and stapes, and this bone and tympanic membrane amplifies the sound. 18-time amplification of sound is there, and the amplified sound enters the inner ear. Now in the inner ear, there are hair cells, which is called a sensory organ or organ of body. Hair cells which converts sound signals into the electrical signals. Now this is most important area. So inner ear converts amplified sound signal into electrical signal. When this electrical signal then is carried to the brain, we hear the sound. And based on incoming sound signal, different electrical signals are generated. Now here, when there is any fault in the inner ear hair cell, a mechanism of converting sound signal into electrical signal, then patient don't hear the sound properly, and we call it as a sensorineural deafness. Sometimes some damages are such that it doesn't affect hearing, but the damaged hair cells, it is in such a stage that even though there is no sound from outside, it spontaneously, meaning by on its own, spontaneously goes on generating electric current continuously. And this is a base of a tinnitus. So even though there is no external sound in the atmosphere, inner ear hair cells continuously generates electrical impulses and patient hears some sound, and this is we call as a tinnitus. This is just a video. Because of visual effect you will understand. When sound comes, eardrum vibrates and amplifies the sound 14 times. This sound is still further amplified by ossicular change. These are three bones, malleus, incus and stapes, and this amplified sound enters the inner ear. And when it enters the inner ear, there is a fluid in the inner ear and hair cells inside the inner ear. So that fluid vibrates, and ultimately this hair cell vibrates, and because of which electric current is generated in the root of our hair cells. And that electric current that electric current, when reaches to brain, we hear the sound. So this is just to keep a visual memory in your memory. Now, at the outset, I am really thankful to our association and our office bearers, and also Lincoln Pharma, to organize this small CME and allowing me for presentation on a management of tinnitus, and mainly focusing on one newly launched drug called Kerovarin. In addition to Kerovarin, I am going to tell you some points about as a whole management of tinnitus also. But main focus will be on Kerovarin. When we say that it's a promising drug, immediately question comes to our mind about reliability of that claim. And so it is necessary for all of us to know that on what concept it is said that it can be used in a tinnitus. What is a proof of that concept? Whether clinical trials are done or not, whether it's only one center trial, multi-center trial or multi-country trials are done or not, what are results? And based on such evidences only, we usually accept some molecule to use in our patients. So today, here I want to tell you about some such certain proofs, concepts, and how I'm using it. I have used in a clinical trial, and what were the results. And because as it is claimed, it's the world's first effective treatment for tinnitus, but only for a cochlear synaptic tinnitus. And we ENT surgeons know the different types of tinnitus. I won't go in much depth of it, but we all know that in the patients of tinnitus, cochlear synaptic tinnitus is in percentage-wise, more than 80% of the patients are of a cochlear synaptic tinnitus. So if it really works the way it is claimed, it is going to give excellent benefit to our patients of tinnitus. I am very happy particularly with this launch because I was directly involved in some of the research activities in a clinical trials. And in 2002, once I understood this concept of carotid, how it works and everything, which I am going to tell you now to you, I conducted a double-blind randomized placebo-controlled study, particularly for the purpose of checking its safety, efficacy, two things, and in cochlear synaptic tinnitus patients. And this was done in 2002. And I was satisfied with the results. My results were at par with the results of other centers in our country. It was also at par with clinical trials conducted in few of the European countries also. Now, if we really want to understand how it works, on what concept this molecule came in existence, it is necessary to understand the primary things about tinnitus. We all know that tinnitus is a perception of a sound without any external acoustic stimulus, which may or may not disturb sleep or hearing. Normally, our patient comes to us when it is disturbing their routine day-to-day life, like a problem in hearing, it disturbs sleeps, or it's in annoying situations. Otherwise, usually our patient doesn't come. Then also, if you look to the incidences of tinnitus are very high. According to one survey, 40% of the normal population, in one or other temporary or a little longer time, they have experienced some such sound in their ear. And 8% of the patients, they complain about it. 1% of the patients, they are keen for treatment. And they are really disturbed because of such sound. To the extent, in a span of my practice of 22 years, I came across at least 7 to 8 patients who did suicidal attempts because some clinicians said, "Yeah, you'll have to live with this, and there is no solution for it." Believe me, this is really a frustrating experience for patients. And of course, we were also feeling a frustration when our loyal patient wasn't coming to us. They follow our line of medications, they come back that there is no improvement, and we also feel frustration. So if really something works, nothing like that. For that purpose, we need to classify tinnitus. Basically, for the understanding the classification, objective tinnitus, where other person can also hear, and we are not concerned here about it. Subjective tinnitus, it is a auditory origin or a non-auditory tinnitus. This is very non-auditory tinnitus, very rare cases. In this span of 20 years of practice, 4-5 patients I came across. Gaze evoked tinnitus. Patient complains typically that when a particular gaze, when he looks, he feels some sound in the ear. And cutaneous evoked tinnitus. Recently also I came across a patient who says, when I scratch my skin, I hear tinnitus sounds. Mechanism still not understood. But we are concerned with this auditory tinnitus. Myogenic tinnitus is a middle ear origin tinnitus. It is because of a tonic and clonic flickering of a muscles of the middle ear. Cochlear synaptic tinnitus and the retrocochlear, we all know about it. But the important thing is incidences of cochlear synaptic tinnitus, 90% of the patients. Now, this is just to show origin of tinnitus because of a tonic or clonic spasm of muscles of the middle ear. Now, cochlear synaptic tinnitus. We will be focusing more on it. Various hypotheses where this is a research molecule by Professor Klaus Ehrenberg, professor of University of Vienna, Austria. In 1992, he proposed first causal hypothesis to why the tinnitus is. I'll explain to you in detail. And he did animal trials and proved, and he presented and published proof of his concept of a
receptor mechanism in 1994. And based on this proof, the exact cause of tinnitus, why tinnitus appears in a damage in a cochlea is understood. And based on this, he proposed, he thought of one molecule called carovarin that it can act to treat this, correct this fault in the cochlea. And he published a paper in 1995. I think the papers of these reference and websites and papers and references of different journals where it is published, is the list is given by this company to all of you. It's given or not? Huh? So they are going to distribute so that if you want to study it in detail, all references, publications, everything, because it is published in all international journals.
Excuse me, I want to ask how you do on animal trial. This is a sensation.
I'll explain. I'll explain. Based on this possible effect of a carovarin in 1997, a double-blind first pilot study was done in Europe, and then other trials were done. Papers were published, and I was convinced after result of this in 2001, for Indian patients, I conducted a clinical trial. Now the concept is, because we all know when sound reaches to the inner ear, when sound reaches to the inner ear, basilar membrane, outer hair cells, and tectural membrane, it does shorting of intensity and frequency of the sounds and pass the information to the inner hair cells. Now, in inner hair cells, this is a simplified diagram. Later, I think, show it. We can record this later on also. This is a simplified diagram of inner ear hair cells. That when sound signal reaches to the inner hair cells, the sound energy is converted into mechanical energy is converted into a chemical energy in the form of a release of a glutamate granules. Glutamate granules are there in the vesicles of our inner hair cells, and when these glutamate granules are released, which is the quantity of glutamate granules and quality of glutamate granules depends on a type of sound stimulus, and based on that, these glutamate granules comes in a synaptic cleft. Now this synaptic cleft, post-synaptic membrane, there are specific receptor, NMDA and AMPA receptors are there, where glutamate granules reaches to receptor, and ultimately it generates an electrical impulse because of an interplay action between receptors of a post-synaptic membrane. And this electrical impulse reaches to brain and we hear. Now this is a physiological state where when sound stimulus comes, electrical impulse is generated. But in certain pathological conditions where inner hair cells are damaged, in this situation, when sound signal comes, glutamate granules which are released are either in a large quantity and are in a different characteristics, and what we claim it as a toxic granules. And that toxic granules, it reaches to the post-synaptic receptors, post-synaptic membrane receptors, and electric impulse is generated. But the characteristics of these granules is different than normal physiological hair cells characteristics. Normally, glutamate granules comes here, it creates an impulse interplay between because of interplay action between receptors, electric impulse is generated, and immediately that granules gets dislodged from the receptor side, and that receptor is now ready for receiving new sound to come. In a pathological situation, toxic granules, it remains adherent to the receptors. It has got a high affinity for these receptors, and because of that, that interplay action is disturbed in a post-synaptic cell, and a continuous generation, it creates a situation called spontaneous depolarization state. And so spontaneously goes on releasing electrical impulse, and that is what patient hears as a tinnitus. Now, why these hair cells are damaged, or what is the mechanism of the inner hair cell damage? We all know that loud overexposure to the noise, local or general circulatory disturbances, metabolic disorders, because of which toxic products comes in a circulation and they may damage this, inflammatory reactions of a middle ear, particularly of a viral etiology, and a perfusion of a toxic product of that inflammatory reaction goes to the inner ear. some traumatic situations, hypoxic situations because of aging also. Now here when we talk about aging, this is very important theory which we today accept as a free radical molecule, and based on which today we prescribe not only this, so many other conditions we started prescribing antioxidants. When different metabolism occurs in our body, like carbohydrate, protein and fat metabolism, during that metabolic process, a free radical atom which possesses an extremely reactive unpaired electron are generated, and these are generated because of inhalation of polluted air and polluted variable oxygen and hydroxide OH radicals in the air. So when you inhale polluted air in aerobic respiration, these type of free radical atoms are generated, and this free radical atom possesses extremely reactive unpaired electron, and this free radical, it when goes in the body and comes in contact with other cells, these, because they are highly reactive unpaired electrons, they picks up electron from a normal atoms of a normal cell, and because of that cells are now damaged. And that if this atom is this electron is picked up from proteins like DNA, then it's a long-term damage, and this is a concept because of which aging is there. And so to prevent aging, we started prescribing antioxidants. The process like cataract and arteriosclerosis and even cancer is claimed to be because of this existence of free radical, and this is one of the main reasons of tinnitus, damage to the inner hair cells in a patient of tinnitus. And so this is just to demonstrate how this free radical picks up free electron from outermost orbit and creates such radical oxygen species which is dangerous for body as a whole. Now this is as a whole concept of a synapse in a cochlea, how it damages, and this is a small video which will clear the as a whole idea of this. Synaptic cleft is an area between end of one nerve and the beginning of another nerve, and end of one nerve, this bulbous part, we call it as a knob, and when this synaptic cleft, it is of more than a 20 nanometer size, it is known as a chemical synaptic cleft. So in some of the area all over the brain, the certain clefts are chemical clefts, and neurotransmission is by release of some chemical. In a presynaptic cell, when a presynaptic cell, this nerve contains vesicles which contains glutamate granules. Glutamate is a neurotransmitter, in fact, all over the brain. When a stimulus or impulse comes to the nerve of this cell, ultimately based on a type of a sound stimulus, number of granules or what type of granules are released, and these granules reaches to the receptors on a postsynaptic membrane, and it stimulates it, and because of interplay between that, electric impulse is generated, and this molecule glutamate granules is dislodged from the receptor side, and electrical impulse is generated, reaches to the brain, and we hear the sound. But in pathological status, when this stimulus comes, glutamate granules are released, it reaches to the receptors on a postsynaptic membrane, remains adherent to it, generates electrical impulses, and instead of getting dislodged from there so that receptor receives new signal, it remains there, and so cannot receive new sounds, and goes on generating electrical impulse in the postsynaptic cell. And this is how tinnitus is there. Now this is the reason, and such situation occurs in select if it occurs in all the hair cells, patient will have associated deafness. But if it is in select or some of the hair cells, patient's hearing may remain normal, but there will be a problem of discrimination because some of the sounds are missing, and so there is a major problem of a discrimination. They hear the sound but don't understand the sound. So Now, then how to solve the problem? How to remove these toxic granules which are adherent to the receptors on a post-synaptic membrane. Because of structural, during that days, because of a structural similarity of a molecule called caraverin, which was in use for colicky pain in the abdomen since 1960s in the European market, and its structural similarity was exactly to that of caraverin. Professor Klaus Edenberger came with a hypothesis that perhaps the caraverin may help if it has got a certain affinity for our post-synaptic receptors. Now here I'll bring your attention that in a post-synaptic membrane there are specific subtype receptors called NMDA and AMPA. Specific type of receptors are there which are not there anywhere in the nervous system other than cochlea, where this glutamate was active. Now caraverin, being a structural similarity, they thought that this perhaps may act, and they conducted several trials, and based on which, what happens when you inject caraverin intravenously with a certain specific method, when this caraverin reaches here, caraverin has got a stronger affinity for a post-synaptic receptors called NMDA and AMPA receptors. So it forcefully dislodges the glutamate granules from this side, and then itself also gets detached from the receptor sites, and these receptors now are available for new sound to come. So it is a situation like ventricular fibrillation. In a ventricular fibrillation, heart just flickers, doesn't contract. If you give a just big thrust on a heart, it stops for a moment and starts functioning with a normal beats. Same way, caraverin here comes, it forcefully, because of high affinity to the receptor, it forcefully dislodges the toxic glutamate granules and comes in contact with it, it gets dislodged and receptors are again starts functioning normally. And this explains, so when first time it was launched, the question was, how a person who is having tinnitus since years together, with a one shot of injection, how somebody can claim that one shot of injection give a permanent cure to this? So this is
to simplify and remember, this sound signals carried to tympanic membrane. This is a middle ear and this is a inner ear. Now here inner ear, when damage is there, patient persists persistent sound called tinnitus. Simultaneously, some conditions where middle ear is damaged, sometimes patient hears some type of sound which is mimicking to the tinnitus or a ringing sound inside the ear. So this will come in a type of a tinnitus classification. We call as a middle ear tinnitus or a myoclonus tinnitus. All of you know myoclonus means because of a muscle movement. There are two muscles here in the middle ear, and tonic and clonic spasm of the muscles. Sometimes every one of you must have experienced that some body of the muscle just go on flickering. Similar flickering if sometimes for no reason, and that similar flickering if there are two muscles inside the middle ear, and flickering of that muscle sound also gives flickering sound. We call it as a myoclonus tinnitus. Caraverin is not for this type of tinnitus, but fortunately or unfortunately, the incidences of myoclonus tinnitus is very low. Same way, the connection from our inner ear to the brain. This is inner ear we call as a cochlea, and behind the cochlea, whatever connection up to the brain, we call it as a retrocochlear. So any fault in the retrocochlear, when I'll explain the mechanism why tinnitus is here, how tinnitus appears, you'll understand that the similar type of damage is possible in the connection between inner ear and brain, and that is called like retrocochlear. So some of the tinnitus, some of the ringing sound in the ear are because of pathology or a damage in the retrocochlear area. Caraverin is not for that retrocochlear lesion also. But again, only 5 to 6% incidences, patients of out of tinnitus patients, 5 to 6% are only because of a retrocochlear. So majority, whatever patient comes to us, 90% of patients are because of a cochlear tinnitus. And in the cochlea, organ called synapse, which is at fault, so we call it as a cochlear synaptic tinnitus. So 90% of the patients, we are able to give some solution here. This is just to show two muscles of a middle ear which are at fault. So we are not much concerned here. But the classification, can you see these white colors or not? Okay, fine. So this is just to classify type of tinnitus which you should know. Tinnitus could be objective. There are some conditions where patient hears a sound, he complains a sound inside the ear, meaning by tinnitus. And if you go near, keep your ear near the patient's ear, you can hear. You keep a stethoscope, you can hear. And these are because of some vascular anomaly, blood vessels or blood vessel tumors at a floor of mouth, we call as a jugular tumor, and some similar anomalies. Patient hears a sound because of a heavy blood flow there, and that hissing sound also is known as a tinnitus, and we call it as an objective. The percentage is less than 1, very less, rather 0.1%. And so we are not concerned here about objective tinnitus. Scaroing has no role, it is a surgical line of treatment. This is just to know about tinnitus. Subjective tinnitus, where patient hears the sound, but examiner or other people cannot hear the sound. That is called subjective tinnitus. And as I told, it is because of we classify it as an auditory tinnitus and non-auditory tinnitus. Non-auditory means ear system, which I explained you is not concerned. These two type of tinnitus, gaze-evoked tinnitus and a cutaneous tinnitus. Just for your knowledge, the patient complains that whenever I scratch somewhere on the body, I hear a tinnitus sound inside the ear. And gaze tinnitus means particularly patient complains that when I look to the right, my gaze is to the right or to the left or some particular position, he hears a tinnitus sounds. The mechanism is because of some cross-fiber connections inside the brain. Incidences are very less, to the extent in span of 22 years, I came across about 4 or 5 patients. So these incidences are very less, but this is just for your knowledge. Now more events are here, and percentage you can see here. Middle ear tinnitus or a myoclonus tinnitus, cochlear synaptic tinnitus is a 90% in retrocochlear tinnitus. So no need to go in detail. Where we are targeting is the highest incidences, and today fortunately we can give benefit to such patients who are in trouble. Now understand the mechanism so that confidently you can explain if required, or at least confidently you can say about your problem. As I explained, when sound reaches to the inner ear, this is the structure of a hair cell, inner ear hair cell, inner hair cells. When sounds, as I explained in that video, that because of a movement of a fluid, these hair cells vibrates, and ultimately this mechanical energy is converted into a chemical energy by these hair cells, and these chemicals, what we call as the glutamate granules, are released. Now, number of glutamate granules released and concentration all depends on a type of sound stimulus. So, which each different type of granulations, different amount of granulations are released, and that is a reason we can differentiate an innumerable type of sounds. These granules, when reaches here, now this is called a synaptic cleft. Synaptic cleft is an area where one nerve cells ends and another nerve cell starts. A cleft-like junction we call as a synapse. And on a post-synaptic membrane, this is a pre-synaptic membrane, this is a post-synaptic membrane, there are receptors. Now this is in fact a one cell and a lipid membrane where these proteins are there, and these acts as a gate. When glutamate granules are released in a synaptic cleft, they reach here, and because of that glutamate granules, a specific type of interplay is generated and specific type of electrical impulse is generated based on what type of sound stimulus is there. So based on a sound stimulus, electrical impulse is generated, and that electrical impulse is carried to the brain. So now this is normal functioning of an inner ear cell in a physiological state. Now, for some or other reason, if these hair cells are damaged, meaning by it's a pathological conditions, I'll tell you different reasons for such damage. But if this is damaged, when sound signal comes, the glutamate which is released, either it is in a large amount or it is a toxic type of granules which are released. Little perverted glutamate is released. Now we don't go in depth of it, but when this toxic granule comes, what it does, it comes here and it generates electrical impulse of a little odd variety. Because of its toxicity, the interplay between this receptor is disturbed, and this disturbed impulse interplay, it generates such a status that continuous depolarization. Normally in a physiological situation, a glutamate when comes and attaches to the receptor, electrical impulse is generated, and immediately these glutamate granules gets dislodged from the receptor side, and that receptor now is waiting for another signal to come. Now in a pathological situation, these glutamate granules, it is attached here, it disturbs the interplay of electrical impulse generation. This is one. And second, it is so toxic, it remains and has got a highest affinity for it, and it remains adherent to the receptor. So that receptor is now not available for the next signal. And this is the reason why some patients also have a hearing trouble along with it. Now this damage is not for all the hair cells. Those hair cells which are not damaged, they go on hearing the sound, and some hair cells, they don't generate sound signals. And so most of the patients, if you take audiogram or a hearing level, they say, okay, we hear properly, sound we hear, audiogram is normal, but they have a discrimination problem because some of the signals are missing. And so they don't understand the sound. They hear the sound, but they have a problem of discrimination. They don't understand the sound. And because these glutamate granules blocks this continuous depolarization state, we call it as a stage of a spontaneous depolarization. So spontaneous depolarization occurs here, situation occurs here, and this cell continuously goes on generating electric impulse, and so patient hears a sound. This is a list of various causes which can damage, and particularly, for example, sudden exposure of a loud sound, a bomb blast suddenly occurs here which damages my inner hair cells, and this starts generating toxic granules. Same way, local or general circulatory disturbance, because of any other reason in the body, blood circulation is reduced there. Metabolic disorders means so many diseases in the body like diabetes, hypertension, etc. And product of that metabolic diseases comes in circulation and damages inner hair cells. Some inflammatory reactions, meaning by middle ear, inner ear is very near to middle ear, so the ear discharge or chronic suppurative otitis media and so many other ear conditions, that inflammatory reactions and chemicals produced because of that perfuse through the membrane to the inner ear and may damage that. Traumatic conditions, hypoxia, low oxygen level, if patient often on stays in a low oxygen level, and because of aging also sometimes for no reason that hair cells are damaged. So these are the reasons. Now here I would like to explain one more important thing, is autoneurotoxicity because of a radical oxygen species. This is generated, what we talk about, a pollution, and this can be explained by because of a pollution in the atmosphere, radical species are generated, and which comes in an inhalation. When we eat food, glucose, protein, carbohydrate, proteins, lipids, and metabolism occurs in a body for a generation of energy out of that food products. That during that metabolism, because of such radical species which comes during inhalation to our body, it damages or it causes little deviation in a metabolism, and a free radicals are generated out of that metabolism. These free radicals, which are the atoms which are having a highly reactive unpaired electron, and that highly reactive unpaired electron molecule or atom, it comes in a circulation, it goes on damaging different cells of the body because they have a highly reactive unpaired electron. It picks up, it satisfies himself by picking up electron from other atoms of the body, and that atom now becomes are damaged. And this is in fact a basic and accepted theory worldwide for cataract, aging process, or a cancer. So this is worldwide accepted theory, and based on this, I think pharma people, you must be knowing, we are prescribing antioxidants lot. antioxidants are based on this concept, and you should understand. And this, why I am explaining that this is perhaps one of the major reason of damage to the inner hair cells. And so, with this concept, in addition to carrageenan has also got an antioxidant action. You can, if you have a product like antioxidant, simultaneously you can promote such product as a additive therapy for a patients, or those patients who are not successfully giving full results with the carrageenan, antioxidants can also be given. And so, which helps in curing tinnitus, also prevention of aging, also prevention of so many other conditions. And so, this was an important concept to explain. This is just to explain how these radical species of oxygen, it picks up the electron and causes damage to the normal hair cells, and aging occurs, or a cell damage occurs, which results in a cataract or a disease like aging or even cancer. So this was little deviation from this. Now this is summary, a small video to explain this summary so that you can memorize it very easily. This is a nerve ending. One nerve ending, another nerve ending, and a synaptic cleft. A space between this is a synaptic cleft. When one nerve ending is there, the end is bulbous. We call as a knob. When the space in a synaptic cleft is more than 20 millimeter, it is called a chemical synaptic cleft. Here there are different mechanisms, but chemical synaptic cleft, where chemical comes here and action takes place. And this is presynaptic cell, that was postsynaptic cell. This presynaptic cleft contains vesicle and has a granules in it. Whenever electrical impulse comes here, in response to electrical impulse, this granule is released into the synaptic, from a synaptic knob into the synaptic cleft. It reaches to the receptors of a postsynaptic membrane, and receptor, it generates electrical impulse. It gets dislodged immediately, these granules, and now receptors are waiting for another signals to come, and your electrical impulse is generated and goes to the higher center. But in a pathological conditions, because of reasons I already explained to you, a toxic granule comes here. It reaches to the receptor sites, generates electrical impulse, but disturbs the interplay of electrolytes. It remains an adherent to this place, and this is the reason for the patient receives continuous sounds of a tinnitus. So it remains there. Now I think theory you may perhaps forget, but this picture memory, this will be good for you. Now then, what to do? So in 1992, Professor Klaus Edinburgh at Vienna University, Austria, he suddenly came to know about chemical, how these sounds are transmitted. So about glutamate he came to know, and glutamate, toxic glutamate, is a cause for it. A drug called carrageenan is in use for a colic pain in abdomen in European countries since 1960s. And suddenly he came to know about a structural similarity of a carrageenan and glutamate. So both have a similar structure. So carrageenan is a drug used for since 1960s. So it was used in human being safely without any complications. Structural similarity. So he thought a hypothesis that perhaps if this carrageenan has got an affinity for this receptor more than a glutamate, perhaps it may work. So he did animal studies, and animal studies he proved his concept that this carrageenan has got a strong affinity for the receptors of a postsynaptic membrane. So if carrageenan is pushed into the body, when it reaches to the synaptic cleft, it forcefully removes the toxic glutamate from the receptors. It attaches there, stimulates receptors, another impulse starts, and immediately, like a normal glutamate molecule, gets dislodged, and now that receptor is free to receive new sensation. So this in animal animal studies he proved. So proof of concept is there. Now these things, this concept in detail, I think you will be giving to them in the form of a printout. So when you go to ENT surgeon, you can always say, when the first presentation, you can always say that this is a new molecule launched which is effective in cadaverine, and this is based on an absolute scientific way. This is derived since after a long study of 16 years from 1994 to 2010, 2011, a 16 year of span, various studies were done. In fact, preclinical trial, animal studies, and a proof of concept is there in an article. And if you go through it, it will tell on its own. Different sites are also there. So they will be convinced. You don't need to explain all these things. You know now what it is and what is the content of that article. So one article of explaining concept of cadaverine, how it is invented, should be there. Now this cadaverine, when it goes to the, it is from a circulation, it reaches here, it forcefully dislodges it, and it starts functioning very well. To simplify still more, it is like a ventricular fibrillation of a heart. You may not be knowing it, but all of you must be knowing that if heart stops, we give a thrust on a heart and it starts. In fact, this is a situation where heart contractions are weak because of electrical disturbances. So instead of full contraction of a heart just goes on flickering like this. This is called ventricular fibrillation. In this situation, if you give a thrust on a heart suddenly stops and starts functioning with a normal contraction. It's an almost similar mechanism that cadaverine comes here, it forcefully detaches the toxic granules, and for a moment there is a block and it starts functioning normally because this cadaverine dislodges from this situation. So based on this concept, various studies were, I'm sorry, I cannot read. Okay, this is about what I explained here, pathological things, and based on this, cadaverine molecule came in the market, and Professor Klaus Seidenberger, he did a clinical trial in his country. He published that paper, and a copy of that paper will be given to you, which you can always give. That in a clinical trial also he proved. So he gave a concept of a tinnitus, he gave a concept of a cadaverine, and then a clinical trial also he performed. And all these three proofs are with this article. And based on this, I conducted a clinical trial for Indian patients because the results may vary with a different population. In Indian patients, initially one central trial and then multi-centric trial were done, and the results are same as what they claim, and the results are excellent. If they wish, we can give a clinical data results of these patients. One sheet of a clinical data also will be there, which prove that results are also excellent with this. But simultaneously, we should also explain to them that a word of caution: it's not a magic, or it's not a magical drug that everywhere, anywhere you use and you give a results. It is, as I said, it is effective only in cochlear synaptic tinnitus, and overdose may increase, even though it could be temporary, but it may increase the tinnitus and patient will blame you. So be careful. So perfect diagnosis of cochlear synaptic tinnitus is necessary, and a proper method of infusion of tinnitus, infusion of cadaverine is necessary. So when you go to ENT surgeon, the second step, you can always say that it is an excellent drug, but it is indicated only in cochlear synaptic tinnitus. And so we have a manual how to guide how to diagnose perfectly the cochlear synaptic tinnitus. So just give that manual. I think in user manual
user manual.
So in one manual, one printout will give you where how to diagnose cochlear synaptic tinnitus is there, and one user manual how to use cadaverine is also there because overdose increases it. Again, coming to the classification, as I told, how to diagnose out of this, we need to rule out myoclonus tinnitus, cochlear synaptic tinnitus. Rest of the things are rare and it's can be diagnosed on its own. And this is a list of investigations. And these details we are going to give you in a printout, which always you can guide. And simultaneously, at the same time, you can say that some of the investigations are not mentioned in our textbooks, which are invented by Klaus Serenberg. Some of the investigations and interpretations by experience I introduced in it for diagnosis. And this, if they want, you people can insist them that you can organize a CME where these explanations will be there from experts if they want. Otherwise, it is there mentioned in their literature. But in a CME, again, such with the presentations, they can be trained for how to how to perform a diagnosis of cochlear synaptic tinnitus. So, for you, going in such detail is not required. But with this, you can definitely diagnose cochlear synaptic tinnitus and how to use it. So, in a user manual, but you should know that it is available in the form of an ampule which contains 8 ml solution containing 160 milligram, and it is to be infused in a pint of 100 ml of a normal saline at a rate of 2 ml per minute. So, not like a bolus. Suddenly, if somebody pushes it, a dangerous complication can be there. And so, in a word of caution, you should always guide ENT surgeon that there is a specific method of use. Otherwise, it may create some chaos. And the dose, how much to be injected through a pint. So, this is very important that till the therapeutic effect is achieved or total solution is injected. The explanation for this is